In this video we will discuss about FAS ligand and FAS interactions controls the persistence of CAR NK cells. So first what they did is they check FAS expressions in the activated NK cells, and they are able to see that in the activated NK cells the FAS expressions are higher, and these things are quantified in this panel B also for showing the reproducibility. And when they compare it between the T cells and NK cells, they can see that T cells has high transduction efficiencies compared to the NK cells. And thereafter they wanted to check the CAR T effect. That is, they introduced the CAR that we discussed in previous videos without any this FAS, or the control CAR which has CD19 and CD28, and the CAR that does not have these FAS signalings. And what they are able to see that in the absence of FAS-L ligand, all of them shows almost no presence of this annexin 5 or active caspase, which is the marker for apoptosis. But once they are activated, they can see the rise in the cells with active caspase 3, 7 and annexin 5, and the rise is significantly higher in the case where CAR is introduced but at delta FAS or FAS signaling pathway is not disturbed, whereas when in the non-introduced the value is low, around 10.5, whereas in the case of FAS signaling has been knocked down, the value is low, around 3.4, indicating that the FAS signalings, once it is get triggered or through this FAS-L ligand, it can cause the apoptosis of the cells. Thereafter they wanted to check what kind of FAS are present in the cells, and they are able to see the bands for both the soluble and the insoluble FAS, both in the case of control and in the case of this ECF, that is where FAS is not knocked down. And when the FAS is knocked down, they do not see any signals, or they do not see any signals for the FAS. This can this can be used as a further evidence to conclude that their knockout is sufficient. Thereafter, what they did is they co-culture the cells, these CAR T cells. They co-culture the CAR T cells with this K562 CD19 cells that has either FASL wild type or FASLG knockdown, and then they observed this TEGFR or and TLNGR ratios. So in the case where K562 CD19 is used, as it helps in the activations of this, this NK cells, as CD19 will interact with the CD19 antigens that are expressed on the CAR NK cells, CAR transduced NK cells. So in the case where FASLG is wild type present, they are able to see the rise in signals for TEGFR upon TLNGFR. TEGFR means that cells that in which FAS is the CAR that is introduced does not have this FAS construct, or FAS has been knocked down, so they can see the EGFR signal goes up in the case where K562 has the FASLG wild type, whereas in the case where K562 CD19 has the knockout FASLG, they do not see a significant rise in the ratio of TEGFR to TLNGFR. So indicating that in the case of knockdown, the TEGFR signals does not get activated because of the disruptions in the FASL signalings. But when the cells are not activated, they do not see any significant changes in both the cases. Now they wanted to check if they knock down this FASLG in the NK cells, whether it will has any impact on the FAS. This is similar to what we have seen in the previous videos. So the results that are shown here shows the similar results, that is in the case of this EGFR and when the wild type CAR is used, they see a decrease in the FASL, decrease in the survival rate of the, and they see a decrease in the tumor cell survivals, a number of tumor cells survived are less, but when they use the knockout, they also see the decrease in the survivals, and the difference is not significant, but even when they use it at different effective to target ratios to from about 1.1:2 to 1:16. So this indicates that when the FAS-L signaling is not been disrupted, it does not when the FAS-L signaling is not been disrupted, as in the case of this wild type with THFR 1920, it doesn't affect the anti-tumor efficiencies of this CAR construct, and it is similar to the case where FASLG has been knocked down in the NK cells and the CAR is introduced with the FAS-L signalings. Thereafter, they do the animal studies where they transduce NK cells and also transduce these CAR T cells without disruptions in the FAS, and the CAR T cells with the FAS signaling has been knocked down. And before that, they introduced the Raji B cells into the mice, and after 4 days they introduced this CAR construct, and what they are able to see that this TEGFR to 2LNR increases, and the reason is because in the case of FAS is not present, in the case of this FAS is not present, the apoptosis will be lower, and in the case where FAS signaling is not been disrupted, the apoptosis will be higher. That is the reason the TEGFR is high. TGFR to TLN GFR is signaling rate is high because of the death of the T cells, NK cells that are transduced with THFR in 28, in which FAS signaling is disrupted. So I hope you understood how they validate their previous hypothesis with some other experiments, that is FAS-L and FAS signaling controls the persistence of CAR NK cells. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.